These are your notes for Chapter 9, Part 2, Translation. As you can recall from the transcription section, we were talking about DNA making RNA. In the translation section, we are going to be in this next section of RNA making proteins. So this is how the nucleotide sequence of an mRNA molecule is translated into the amino acid sequence of a polypeptide or a protein. You don't necessarily say that mRNA makes proteins. mRNA makes polypeptide chains, and then those polypeptide chains are what folds in amongst themselves, and the combination of the folding and all that makes the proteins. So you're going to be using this chart in class on, I guess, Monday or Thursday, or you'll be using one that looks like a wheel. And this is your genetic code changed from the mRNA into the different amino acids. There are, I'm just going to read it. Here we go. In an mRNA, the instructions for building a polypeptide chain are RNA nucleotides as A's, U's, C's, and G's, run in groups of 3 called codons. There are 21 codons for amino acids, and each of them is read to specify a certain, there are 61 codons. Sorry if you did that wrong. 61 codons for amino acids, and each one of them is read to specify a certain amount of amino acid out of the 20 commonly found in proteins. AUG specifies methionine and also acts as a starting codon or a start codon to signal the start of protein construction. So archaeobacteria, one of the reasons why they are considered most like Animalia cells in eukaryotic is that they do also use methionine as their start codon, whereas other prokaryotic cells do not. Where was I at? There are 3 more codons that are very important that do not specify specific amino acids. These are stop codons, UAA, UAG, and UGA, and they tell the cell when a polypeptide is complete. So if these were placed in the middle of an amino acid chain, you might end up with a problem where it stops instead of continuing its construction, and then, of course, structure equals function. Your protein is not going to function correctly. Altogether, this collection of codon amino acid relationships is called the genetic code because it lets cells decode an mRNA into a chain of amino acids. So let's get on into the 3 stages of translation. The first stage, again, is initiation. The second stage is elongation, and the third stage is termination. Sound familiar? Here are your pictures of what's going on. So you've got your mRNA codons, and they code for your amino acids. Again, we always start with meth, and then your amino acids are brought in by tRNA units, and we'll get into that, into your ribosome, and they will join together to make your polypeptide chain as the tRNA brings them in. So we'll talk more about tRNA and the ribosomal RNA later. So let's start with your initiation. In initiation, the ribosome will assemble around the mRNA to be read, and the first tRNA carrying the amino acid methionine, which matches the start codon AUG. This setup is called the initiation complex, and it is needed in order for translation to get started in every single case. So again, you've got your mRNA here, you've got your tRNA here, and its anticodon, means the opposite of the codon, is UAC. So UAC will pair with AUG. That's how the tRNA knows what to bring, and it matches with methionine. So methionine is joined on, and this is your initiation complex right here. mRNA, the UAC, tRNA with methionine attached, and that all is attached to your ribosomal subunit. In elongation, the amino acid chain gets longer. We aren't trying to make anything fancy. The mRNA is read 1 codon at a time, and the amino acid matching each codon is added onto a growing protein. A matching tRNA will bind to the codon. Then the existing amino acid chain, or the polypeptide, is linked to the amino acid of the tRNA via a chemical reaction. And finally, the mRNA is shifted 1 codon over in the ribosome, exposing a new codon for reading. So here's my lovely picture of what's going on. Again, it's got the 5' and the 3' end labeled here. So you've got 3 positions on your ribosome. I've never had to learn those. Maybe you will later on in your biology, the EPA positions. But the P is where it starts its initiation. So you've got your amino acid brought up here. This is your tRNA. The next tRNA will bind in the next slot. Then that chemical reaction will happen, creating a bond between one amino acid and the next. The first one will join onto the top of the second one. Kind of think of like beads in a chain. And then the ribosome keeps moving down and moving down. And then the first tRNA will leave to go collect more amino acids. This one moves to the P phase, and then the next tRNA will come over there. Think of tRNA as like transport trucks that are bringing your amino acids together. They're not really doing too much else. They just bring it in, and then the amino acids through the ribosome will get joined together. Okay, so elongation, we're just getting longer. Finally, we move on to termination, and it's just like as it sounds. We are stopping. So termination is the stage in which the finished polypeptide chain is released. It begins when you hit a stop codon, which is either those three that we talked about, UAG, UAA, or UGA will enter the ribosome. That will trigger a series of events that will separate the chain from its tRNA and allow it to drift after the ribosome. The thing is, after termination, the polypeptide may still need to fold into the right 3D shape and undergo processing, and it will do that in the endoplasmic reticulum, or maybe it gets shipped to the Golgi apparatus before it's finally ready to take its role in the cell. So, parts you need to know. Ribosomes. Ribosomes are structures where polypeptides or proteins are built. Ribosomes are made of proteins and RNA. Get back to my teacher notes. I had to skip around. Each ribosome has two subunits, a large one and a small one, which come together around an mRNA strand. And the ribosome slots where the tRNAs can find their matching codons. It has slots that are there. And the ribosome will also act as an enzyme, which catalyzes the chemical reaction that will link the amino acids together to make the chain. Like I said, the tRNA just brings them in, but the ribosome is what actually will lower that activation energy and cause those reactions to take place. So you can't really build these without ribosomes, because they're not just a point where it can get processed and these things can meet up. It actually is responsible for lowering that activation energy. And if you can remember anything about enzymes, our body processes wouldn't be possible without them, because we would just need so much heat or so much extra electricity or whatever to cause these reactions to occur without enzymes. So they are so very important. And I skipped past tRNA. I'm sorry, I'm going back to this again. tRNA, let's move up here, are molecular quote-unquote bridges that will connect the mRNA codons to the amino acids they encode. At one end of each tRNA has a sequence of nucleotides called an anticodon. I talked about those earlier, which can bind to specific mRNA codons. And again, the anticodon is the complement of that. So these are going to mirror the DNA template strand that we made our mRNA strand from in the beginning, only instead of T's, they have U's. And the other end of the tRNA carries the amino acid, which is specified by the codons. Now remember, let me show you this lovely little chart here, because in our next video we'll talk about mutations, but before I do that, go back to the chart, here we go. Remember, there are 61 different letter combinations that only code for 20 amino acids. So you've got the trucks, but they might all be going for the same amino acid. Like ILE, isoleucine, is this one down here, and it's got actually 3 different codes that will code for isoleucine. I don't remember what THR is, but it has 4 different codons that will code for that one. So it's not always a 1-to-1 ratio. It is 1-to-1 for methionine. Think about that. Why do you think that would be the case? We'll actually have to talk about that later in class, because I want to have a discussion as opposed to giving you the answers. But why would it be the case that you might have 4 trucks for this THR and 3 trucks for isoleucine, but only 1 truck for the starting codon? Something to think about. In the next one, we're going to use this, and we're going to talk about mutations and how mutations can drive the evolutionary force of all of nature.